Hello, everyone, and welcome to today's webinar, Overcome Top Titration Challenges. I am Michelle Ashton of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more, please visit www.thermofisher.com/titrator. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speakers, Ricki Hartwell, Senior Product Manager, Gayle Gleichauf, Applications Lab Manager, and Kevin Goose, Senior Product Manager. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Ricki, Gayle, and Kevin will now begin their presentations.
Okay, great. Thank you, Michelle. Do any of these titration challenges sound familiar? As Allison from Villanova University described, the spout detached from the rest of the burette and shattered. Since the burette was now open on the bottom, the reagent poured out and spilled everywhere. It was a strong base, so we had to neutralize it with acid. It was a big mess of reagents and glass.
All right. Here's one. Does this sound familiar? I was doing an elemental iron titration that was about 2 hours of prep before the final titration for color change. Someone walked in the lab and said my name, so I looked up and missed the endpoint. I had to start all over again with the 2 hours of prep. That's from Aaron of the Heritage Research Group.
Well, does this challenge sound familiar? Becky from Aqua Bounty said, at my previous job, I had to perform manual titrations on all incoming intermediate products for production. Let me tell you, sitting in one place for 8 hours a day, watching minuscule droplets fall into a container, desperately waiting for the color change, was worse than watching paint dry. Don't even get me started about when I'd add too much, too fast, and miss the change altogether.
All right, great. Let's talk about titration challenges and how we can help solve them. Have you experienced any of these common challenges with manual titrations? Are you spending a lot of time performing manual titrations? Do you need to complete more testing in the same amount of time? For this titration tip, we will discuss how you can improve accuracy, safety, and lab throughput by automating your titrations. Sample accuracy is critical to lab measurements. You want to get the correct results the first time so you do not have to recheck your procedures, review your results, and potentially repeat testing all over again. With manual titrations, you must add small drops of titrant, one at a time, into your sample beaker. Watch very closely for the endpoint to be reached by color change, pH or millivolt reading, or similar technique, and then record the volume of titrant used to calculate your resulting sample concentration. And with all these steps required for the manual titration process, there are a lot of opportunities to make mistakes and introduce errors into the results accuracy. Automating your titrations greatly simplifies this process as the titrator manages all of these tasks for you. Automating titrations can also improve the results accuracy several ways. By automating titrant additions using high-resolution microliter volume doses, by using an electrode as the endpoint indicator that eliminates the need to interpret or guess when the endpoint has been reached, and by automatically calculating the concentration results for you using the same parameters and procedures every time. Automating your titration also allows multiple cycles to be run on the same sample, so you can make sure your testing results are reproducible with average concentration and relative standard deviation calculations performed automatically, plus the ability to exclude any outlier cycles from these calculated values. Safety is also an important factor in laboratory environments. Manual titrators require the operator to fill the burette with titrant and flush the stopcock, fill the burette exactly to the 0 mark, and make sure the meniscus level is correct each time they need to perform a titration. This process can lead to some fairly dangerous situations if equipment fails or is mishandled. For example, some common issues include if your burette and stand are too tall, you can spill hazardous titrant on yourself when filling the burette. If your burette system is older or isn't stable, your burette may fall off the stand and break, throwing glass shards everywhere. And if your burette tip or stopcock is loose, it may fail to control titrant flow or fall out and spill titrant all over. Automating your titrations can greatly improve safety in the lab. Since the titrator offers a fully automated dispensing system that draws the titrant from the titrant bottle into the burette and then dispenses the titrant in small, measured drops into the sample, all without the operator needing to touch any of the dispensing components. Our titrators also offer additional safety features, including a protective plastic cover around the glass burette to protect it from damage or breakage, and a programmable maximum dispense volume parameter that prevents the titrator from continuing to dispense titrant if the endpoint is not detected. Another key factor in labs is sample throughput. Most labs are looking for ways to perform more testing in the same amount of time or with the same number of operators. Performing manual titrations can be extremely tedious, requiring the operator to stand in one place, watching minuscule droplets drip into a sample container, and diligently waiting for the color change or other endpoint indicator to occur before starting the process all over again for possibly hours and hours of repeated sample titrations. Automating your titrations allows you to start the titration and then walk away from the titrator to perform other tasks or tests. While the titrator takes care of the titrant addition, endpoint detection, and results calculation automatically without any involvement from the operator. Once the titration is done, the titrator will beep, notifying the operator that the results are available and another titration can be started. Another challenge with performing titrations is the influence of the burette accuracy on attaining good results. Are you having trouble with inconsistent titration results? Are you using large volumes of titrant to get clear endpoints? Are you spending lots of money on expensive titrant or waste disposal? For this titration tip, we will discuss how you can improve burette accuracy for better endpoint detection and titration results, plus achieve potential cost savings by using less titrant and less sample, leading to less waste to dispose of. Manual titrations typically use a non-certified Class B or Class A burette with stopcock to add doses of titrant to the sample. The operator uses the stopcock to start and stop the additions of titrant into the sample, often one drop at a time until the endpoint is reached. Once the endpoint is reached, the operator records the total volume of the titrant added to the sample and uses this data to calculate the concentration results for their sample. The precision of these additions, especially near the endpoint, is primarily determined by the operator's skill level, experience, and focus on the task at hand. All too commonly, an operator can allow too much titrant to flow out of the burette into the sample and overshoot the endpoint, requiring them to perform the entire titration all over again. Even highly skilled and experienced operators can be limited in the precision of their manual titration results by the last few drops of titrant, since each drop can vary in volume. Another limitation is the burette's graduated volume marks and stated accuracy. Typically 0.1 milliliters for non-certified burettes all the way to 0.03 milliliters for Class A burettes. This is significant since the final volume of titrant used for the titration is determined visually by the operator, judging the level of the remaining titrant in the burette using the graduated volume marks. To compensate for these limitations, often larger volumes of titrant and sample are used. When using an automated titrator, the titration is performed using a high-accuracy titrant delivery system that controls all titrant additions into the sample and will adjust the dose rate as it detects the endpoint approaching. Once the endpoint is reached, the titrator uses the precisely measured volume of titrant added to the sample to automatically calculate the concentration results for the sample. The operator's skill level, experience, and focus on the task at hand is no longer a factor in the overall accuracy of the titration results, and the possibility of missing the endpoint is greatly reduced. As a result, titrant and sample volumes can be adjusted so smaller volumes are used. As we can see in this chart from the article, 6 Things That Can Go Wrong with Your Alkalinity Titration, Burette accuracy is a very important factor for the uncertainty value of your titration results. This illustrates how using a better titrant delivery system can improve uncertainty from plus or minus 2% down to plus or minus 0.02%. Look at an example of how using smaller volumes of titrant and sample can benefit lab costs. When performing a manual titration to determine the salt content of prepared foods using a silver nitrate titrant, typically about 20 milliliters of titrant is used per sample titration. Using an average cost of a 1-liter bottle of silver nitrate titrant, we can see that the cost per test is about $2 to $4, versus running the same titration with an automated titrator that is optimized to use 2 milliliters of titrant per sample. By using less titrant with a more accurate delivery system and endpoint detection method, we can see that the cost per test has now decreased to about 20 to 40 cents. Plus, the additional cost savings of having less waste to dispose of can really result in some great savings for your lab. Now I'll turn it over to Gayle to discuss our next titration tip.
Great. Thanks, Ricki. And hello to everyone who has joined us today for this webinar. First, I'd like to talk about challenges around color endpoints. So let me ask you, have you ever missed a titration endpoint? Is the color change endpoint hard to differentiate? Are other operators choosing different endpoints and obtaining different results? And do your colored or cloudy samples make it hard to identify the endpoint? These are some common complaints about manual titrations that I've seen and heard in my own lab. What about you? Do you recognize this titration? This is like a titration of an acid with base and phenolphthalein indicator. Phenolphthalein actually has a pinker color. However, if this was the case, would this titration be before the endpoint, at the endpoint, or past the endpoint? Right. This titration would be past the endpoint, especially since phenolphthalein endpoint is the first faint pink color that persists longer than 15 or 30 seconds. Do you recognize this? This is a methyl orange titration. Which color do you think represents the endpoint? The challenge here is choosing the correct color endpoint. If the wrong endpoint is chosen, the results won't be accurate. There'll be a high or a low bias. Decisions made on the basis of these results can be affected. Would you choose the same endpoint as another person doing the titration? The challenge is that different people can choose different endpoints. So when different people choose different endpoints, they also calculate and report different results. That means there'll be inconsistency in the results in the final product, depending on who is doing the titration today. Here's a Ripper titration for sulfur dioxide in white wine. The endpoint is easy to see. We go from almost colorless to a purple-blue color. And for those who prefer red, here's a Ripper titration for sulfur dioxide in red wine. In this case, the endpoint is not so simple to identify. The wine is red and the endpoint is purple-blue. It's so challenging that some lab personnel will shine a bright light through the wine during the titration to help try to make that endpoint easier to see. The challenge, determining the endpoint of a colored or cloudy sample. This slows down our titration, increases the uncertainty of the results, and impacts the precision and the accuracy of those results. To overcome these challenges, we'll use an auto-titrator with pre-programmed methods. When we automate a titration, we can be confident in overcoming these challenges. We won't overshoot an endpoint. There won't be trouble identifying the endpoint. We won't have different answers for different operators, and we won't be confused by colored or murky samples. Instead, our titrations will be repeatable and reliable, no matter what titration, what endpoint, who performs the titration, or what sample is titrated. So converting to an automatic titration is often a simple one-time task, resulting in a saved programmed method that can be copied, shared, and used for all future titrations. Then, with a few touches on the screen, we can run an automated titration from the saved method. Once we start the titration, we can walk away and perform other activities in the lab or at our desk, maybe even grab a cup of coffee. A simple option for automating a manual titration is to use a pre-programmed method that's already available in the titrator. These methods are available in application notes on our website or by contacting your local representative. These application notes contain a wealth of information to help you be successful with your titrations, including the recommended equipment and solutions, the programmed method, sample preparation information, performance data, and tips and tricks for customizing your method and getting the most from your titrator. In the field of water, here are some examples of titrations that are easy to convert from manual to automatic titrations. Alkalinity as total alkalinity or M&P alkalinity, calcium hardness, total hardness, chloride, and Winkler DO. Here are some examples of food and beverage titrations that are simple to automate. Sulfites in wine, acidity of orange juice and other beverages, salt in prepared food, and sodium in canned vegetables. And here are some examples of chemical petrochemical titrations that can be automated, including acid number, or TAN, base number, or TBN, mercaptans, and chlorides. All right. So have you ever noticed that an old container of titrant gives different results than when it was new? Does your titrant change color over time? Have you noticed precipitation in your titrant bottle or burette? Does your titrant evaporate and lose volume over time? If any of these changes happen, it means that the titrant concentration is no longer the same as the value printed on the bottle or the certificate of analysis. Any titrations that we do perform with this compromised titrant will not give us accurate results. So let's talk about titrants and some challenges we face regarding the integrity of our titrants. True or false? The titrant concentration printed on the bottle or the certificate of analysis is accurate throughout the use life of the titrant. Well, the answer is, depending on the titrant and the storage conditions, that may be false. Here on the right is an example of how the concentration of a potassium hydroxide titrant in isopropanol changed over 5 non-consecutive days of testing in my lab. This is the titrant used for acid number titrations in oil. By the last day of testing, the actual concentration of the titrant in the burette had changed by about 18%. If I had assumed that the concentration of the titrant was still 0.1 normal as stated on the bottle and on the certificate of analysis, my results would be off by 18%. That wouldn't be good. Over time, standard titrant solutions age and can change concentration due to a variety of factors that can affect the titrant integrity, including air exchange. Oxygen and carbon dioxide in the air can interact with our titrant. Evaporation, precipitation of a component in our titrant, exposure to light, or just contamination. This is particularly true for certain titrants. Here are some examples. Base or alkaline titrants, iodine titrants, silver nitrate, thiosulfate, and arsenite titrants, and non-aqueous titrants. When the titrant ages, a certified concentration may no longer be accurate. When the titrant concentration is not accurate, our results won't be accurate. So when increasing the accuracy of a titration is important, look at standardizing the titrant and take steps to preserve the titrant integrity. For best accuracy, periodically determine the exact concentration by standardizing the titrant. Note that it's common to standardize on a daily or weekly basis, but other standardization frequencies may be suitable. Standardizing is as simple as titrating a measured volume or weight of a known standard. The true titrant concentration is then calculated and used for future sample calculations to improve the accuracy and the confidence in our results. The option to standardize is available in each programmed method. When activated, the home screen of that method will then show the choice to standardize whenever desired. When standardizing, we can run one standard titration, or we can run replicates up to 5 times to increase the confidence of our results. The titrator will then calculate the true titrant concentration and the RSD of the replicates so that we don't have to. This true titrant concentration will automatically be saved and used for subsequent titrations with that method. In this way, we are achieving better accuracy with our sample titration results. Some customers may prefer not to standardize their titrant. Instead, these customers can choose to purchase and use certified standardized titrant solutions. In this case, users can edit the titrant section of the method and enter a certified concentration and a titrant ID. Entering the lot number in the titrant ID is a good way to track our titrant data. In either case, whether standardizing or not, take steps to preserve the integrity of your titrant. Use the recommended titrant bottles, caps, and tubing, which are designed to minimize evaporation and prevent particles from getting into the titrant. Protect your light-sensitive titrants. Use a brown or opaque bottle to hold your titrant. Some titrants, such as sodium hydroxide titrants for certain titrations, can be protected from the carbon dioxide in the air. Place a carbon dioxide sorbent in the scrubber tube on the titrant cap air intake. The tinted burette cover automatically screens out UV light to protect the titrant in the burette. And choosing opaque tubing will also protect your titrant from the effects of UV light. By taking these steps, we can extend the use life of our titrant, minimizing the frequency of standardization and increasing the accuracy of our results. Now let's look at some tips to improve our titrations. With a few simple modifications, we can take steps to make our titrations shorter, to do more titrations in less time, waste less sample, save on expensive titrants, and generate less waste. We can customize our method to titrate faster and run more samples. When speed and sample throughput are important, there are different options for maximizing the titration. Stirring faster will speed the titration as long as there's no splashing and no strong vortex created in the sample. I've found in my lab that going from a medium to a fast stir can make a titration go 35% faster. That means that if I used to spend an hour titrating a batch of samples, now it will just take me 40 minutes. Or I could run a third more samples in that hour. Another option is to speed up a titration by choosing a different titration process control parameter. For example, if the control is set to routine, we can try changing to quick to speed up the titration. I've found in my lab that going from a routine to quick process control can make a titration go about 14 or 15% faster. For example, that shaves about 35 seconds off 4-minute titration. Another option is to make use of the predose parameter. If our titration typically takes a known amount of titrant, we can program a predose into the method. When we do that, the titrator will quickly dose the amount of titrant right at the start and bring us close to the endpoint right away. For example, if a titration typically takes about 5 mils, we might choose a 3.5 or a 4 mil predose. Titrator will add the predose quickly, and then only must titrate for the endpoint in the last mil or so. I've found in my lab that adding a predose can shave minutes off a titration time, and I can get a lot more done in a shorter amount of time. And the last tip that I'll discuss is this one. This option is the option to titrate less sample. For example, if the 50 mil sample takes 10 mils of titrant, we can try using 10 mils of sample, which will only take 2 mils of titrant. When using an auto titrator, 2 mils can be added in 0.001 mil increments, and that allows plenty of precision, even though only a small amount of titrant is used. I don't need to dispense a large volume of titrant in order to obtain precise and accurate results. Reducing the amount of sample titrated means a shorter titration and brings the benefit that less sample is required and less of your titrant is required, and will generate less waste per sample. Well, thanks for listening in on these titration tips. I hope you found them helpful. Now I'm going to hand you over to Kevin Goose, our Senior Product Manager, who will provide some great tips for choosing your electrode, calibrating, and caring for your electrode for the very best performance. Kevin, take it away.
Okay, Gayle, thank you for that intro. So moving on in choosing the right electrode, we'll look at how you can achieve accurate results, what produces the fastest response, how will you ensure repeatability, and is it possible to mitigate temperature effects? In the next few slides, I'll go through some best practices to help you get the best possible titrator results. When faster titrations are sought after with improved precision and accuracy, you best know that your electrodes need to be in peak condition to provide the results that you're looking for. An accurate, fast, and repeatable pH electrode leads to fast and accurate pH titrations, so choosing the right one is important. For example, ASTM has written standards for acid number and base number titrations that recommend a specific type of pH electrode be used in conjunction with the test procedures to verify the specified electrode performance. The pH electrodes that we recommend in our non-aqueous pH titration application notes for acid number and base number meet the performance specifications of these ASTM standards. Numerous app notes, as shown on the slide, have been written by Thermo Scientific that specify which selected electrodes would be most suitable for an automated titration. The use of the right automatic temperature compensation probe coupled with one of the most thermally stable Ross electrodes will get you to the results that you're looking for. Please note, for non-aqueous samples, a SureFlow electrode with a spring-loaded flushable open annular junction can easily be utilized to replace the outer fill solution with a non-aqueous fill solution to avoid junction potential bias. Now let's move on to accuracy. In most cases, it is recommended that you calibrate your pH electrode once a day when titrating acid-base samples. If in doubt about the stability of the calibration over the course of the day, read a pH buffer standard in the measure mode that is close to the endpoint pH that you're looking at. If the buffer reads close to the accepted value, at the measured temperature, your calibration is still good. Typically, we're looking to be within plus or minus 0.05 pH units of the standard. In some cases, for example, if the titration technique is an equivalence point rather than a preset endpoint, and the measurement units chosen are millivolt units rather than pH units, calibration is not necessary for accurate titration results. However, calibration still gives valuable information about the performance of the electrode and can indicate the need for maintenance or replacement. Let's look into some added tips. It is always best to calibrate with at least 2 buffers that will bracket your end measurement. Try not to have a pH difference of these 2 buffers more than 3 pH units to help improve accuracy. While some users may wish to do a multi-point calibration, it is always recommended that a multi-point calibration be done in a sequential point-to-point method and to not use the linear regression method. For example, a common practice is to calibrate with a 4 and 7 buffer and to verify against a 6.86 buffer. For our electrodes, when it comes to measuring slope, as a result of the calibration, we typically look to have a slope within 95 to 102% and an offset of no more than 59 millivolts, so we can look at our automatic buffer recognition. Now let's look at caring for your electrodes. Good electrodes require some routine care, just like a car. Flush and refill the filling solution regularly, weekly, or biweekly is usually effective. Store the electrode in the recommended storage solution and change the storage solution biweekly. Or if storing it long-term, place the electrode into the protective cap that originally came with your electrode with some additional amount of storage solution. So for some added cleaning tips, depending on the type of sample analyzed, periodic cleaning may improve the performance of the electrode. The type of cleaning solution used will be influenced by the samples being analyzed. Grease and oily samples, disinfection requirements, and proteins are all examples of required specific solutions for proper cleaning. Please follow the recommendations in your application notes or the pH electrode user guide that accompanies each electrode. Those are the tips that are being covered for caring for your electrode and getting the best accuracy for your titrator needs. And back to you, Ricki.
Great, thank you, Kevin. For our last tip, let's discuss challenges with performing titrations and properly recording titration data, correctly calculating the sample concentration from the endpoint volume, and maintaining detailed records for long-term storage and testing verification. Are you spending a lot of time manually logging your titration data? Do you want to easily calculate sample results from your titrations? Do you need to maintain better long-term data storage records? For this titration tip, we will discuss how you can simplify data recording and calculations and improve long-term storage of your data records. To simplify data collection, we would never forget to record an endpoint volume. Instead of manually recording the endpoint volume after performing a titration, use an automated titrator that automatically records the endpoint volume, in addition to the individual points that compromise the titration. So tabular data can be viewed on the titrator's computer software or in Excel. Plus, automated titrators allow you to view the real-time live graph of titration data on their displays, allowing for immediate verification of titration progress and result accuracy. Another way to simplify data collection, we would always use the correct formula to calculate titration results correctly. Instead of manually calculating the sample concentration from the titration results, use an automated titrator that automatically calculates the sample concentration from the entered parameters. Plus, on the automated titrator, electrode, titrant, and titration setup parameters can be saved as a method, so the exact same settings are used for each titration. These methods can be transferred between titrators for consistent procedures to be used on multiple titrators or multiple labs. This way you can save time with repeated titrations by running the exact same parameters each time without having to re-enter information. Our next way to simplify data collection, we would always ensure the data is properly saved for long-term data retention. Instead of manually logging the titration results in a notebook or scrap of handy paper, an automated titrator will automatically save the titration results in the data log with time and date stamp. Plus, for long-term data retention, the automated titrator allows you to print data to a compact printer or transfer data files using the titrator's computer software or a USB flash drive with your choice of PDF report files or comma-separated or CSV files for use in Excel. So now you can use your preferred way to retrieve critical data for tracking requirements. Thank you for listening to our tips for improving your titrations. I hope you have discovered some new ideas to help with your individual testing. Let us help solve your titration problems with the Thermo Scientific Orion Star T900 series automated laboratory titrators. We offer 3 dedicated models for frequent routine titrations of similar samples: the Orion Star T910 pH titrator, the Orion Star T920 redox titrator, and the Orion Star T930 ion titrator. Plus, we offer one multi-purpose all-in-one model for occasional titrations of varied samples, the Orion Star T940 all-in-one titrator. For more information on any of these instruments, please visit thermofisher.com/titrator. Thank you again for your time and attention.
Thank you, Ricki, Gayle, and Kevin, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's get started. Our first question is, can you address titrations of very viscous solutions?
Hi, this is Gayle. I'll take that question. That's a good question. So viscous samples can present a challenge during titrations. The success of the titration will depend on the ability to stir the sample effectively. If we can't get a good complete stir, then the reaction will be slow and the electrode response will be slow. So if that's the case, we'll want to consider diluting the sample before we start the titration. If the sample is aqueous, we can look at diluting the sample with reagent-grade water. If the sample is non-aqueous, we can look at diluting it with a titration solvent that's appropriate to dissolve the sample. In either case, we may want to consider then running a blank on whatever we've titrated the sample with. Thank you.
Thanks, Gayle. It looks like we may have another one for you here. It says, Gayle, you mentioned using a certified titrant and standardizing the titrant. Which do you do in your lab?
Yeah, well, in my lab, I've used both approaches, using a certified titrant and standardizing my titrant. So, for example, when titrating an alkaline sample with a certified acid titrant, I've found that that concentration stays quite stable over time. And so therefore, I don't feel that I need to standardize it with any frequency. On the other hand, when titrating an acid number sample in oils, the titrant is potassium hydroxide in isopropyl alcohol. And that's a volatile titrant. And I've found that that concentration does change noticeably with time. So I've found that it makes sense for me to standardize the titrant on a regular basis so that I can keep my titrations accurate as that concentration changes over time.
Thank you. Okay, next question. Can I use my existing electrodes from other Orion meters?
Yeah, this is Kevin. I'll take that.
So, of course, as long as the performance of the electrode that you're using is compatible with the samples that you're trying to assess, absolutely, and as long as the connections are compatible with the titrator itself. So maintaining a healthy electrode that you know is performing well is certainly encouraged and will give you more confidence in your automatic titration.
Thank you. All right, next question. This is a two-part question, so I'll just start with the first part. It's how can we standardize the titrator? And the second part is the titrator approved from Pharmacopoeia?
Okay, so I'll take the first part of that. Our automated titrator has a function built in for standardization. So it just requires a couple parameters be programmed, and the titrator will automate the standardization just like it automates the sample titration. So again, removing all of the detailed technical flow and automating it for you. In terms of approved for Pharmacopoeia, typically it's the process or the method that's approved and not necessarily the piece of equipment. We have a really great team of technical support specialists here that can help you with your individual application. So what we can do is reach out to you after this webinar and find a little bit more information about exactly what your application is and what you're trying to measure, and help find out if our titrator would be a good fit for your samples.
Thank you. Is there a printer option available?
Yes, there is. We have a compact printer available that connects directly with the titrator. It requires no setup. It integrates and the titrator automatically recognizes it. That's our Star A-106 printer. And if you want more information, it's also available on thermofisher.com/titrator.
Great, thank you. Okay, next question. We have the model T9102. How can it be standardized?
Okay, it's a pretty simple process, again, to standardize. When you're under the titrant settings, the default setting is manual entry of your titrant concentration. You just want to select standardization and then go through the parameter prompts, which it'll walk you through everything you need to enter in order to standardize your titrant. And if you have any additional questions, if you need more detail, again, reach out to us and we'll be able to help you. Our contact information is on thermofisher.com/titrator.
Great, thank you. Okay, you mentioned saving money on silver nitrate titrant. We test for salt in food titration. Do you have a kit for that?
We do have the kit for that. We pair our Orion Star T930 ion titrator with a silver billet electrode and give that to you at a bundled discount. We also have an application note and a pre-programmed method available for that particular application, the salted food. So again, all that information is available on our website, and we also have local support teams who can give you more information. So reach out to us and we'll provide that information to you.
Okay, great. It looks like we have one more question here. I've done a number of titrations in the past, and for some reason the endpoint seems to have slowed down and now takes longer. What could be causing that?
Hi. It is probably the case that the electrode could benefit from some maintenance at this point. If the electrode becomes dirty or hasn't been maintained, the response can get slower, and this will slow down the titrator. So if that's the case, we want to take some routine precautions, which is possibly drain and refill the electrode with new filling solution. We probably want to look at how we're storing the electrode to make sure that that solution is clean and that we're storing it in the best possible manner. And it's possible also that the electrode would need to be cleaned, and those instructions are generally found in the electrode user manual
on how to clean and store your electrode.
If they need to refresh the quality surface of the bulb, that would also be done. So, making sure that overall it's slowing down because the response of the electrodes are slowing down because of the aging of the bulb, which can be reconditioned. Or as Gayle was saying, it could be possibly the junction as being clogged and needs to be cleansed, and will also help to speed up the response of the electrode.
Great.
Thank you again, Ricki, Gayle, and Kevin, for your time today and your important insights. We would also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Any questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.